The next lecture will be by Dr. Robert Schuck. Dr. Schuck is a clinical pharmacologist in the Genomic and Targeted Therapy Group at the FDA. In his current position, Dr. Schuck contributes to the regulatory review of investigational new drug applications to effectively utilize genomic and biomarker-based strategies in drug development and regulatory evaluation. Prior to joining the FDA, Robert received his Doctorate of Pharmacy degree from the University of Michigan College of Pharmacy and then completed a PhD in pharmaceutical science at the University of North Carolina School of Pharmacy. Please enjoy today's lecture.
Hi, my name is Robert Schuck. I'm a clinical pharmacologist in the Center for Drug Evaluation and Research at the FDA, and today we'll be speaking about biomarkers of drug effects. I'm going to break the lecture up into three separate parts. I'm first going to discuss biomarker definitions and utility. I'll then go on to discuss the use of biomarkers in drug development, including pharmacodynamic biomarkers as well as surrogate biomarkers. And then we're going to discuss briefly some regulatory considerations in the use of biomarkers. So the first section will be the definitions and utility of biomarkers. So what is a biomarker? A biomarker, which is short for biological marker, generally refers to a measurable indicator of some biological state or condition. Biomarkers are often measured and evaluated to examine normal biological processes, pathogenic processes, or pharmacological responses to a therapeutic intervention. And biomarkers are used in many scientific fields as well as medical practice. So they're used both in science and drug development as well as in clinical practice in the care of patients. Biomarkers are not new. However, the application of novel biomarkers in drug development and regulatory decision making is a current area of focus for many healthcare stakeholders, including pharmaceutical industry, academia, health authorities, and patient advocacy groups. So in addition, over the last decade or so, there have been numerous technological advances in our ability to measure and quantify biomarkers. So this has led to the discovery of many new biomarkers and many new techniques, such as next-generation sequencing, have led to this being a very active area of research currently. And we're trying to figure out how best to utilize biomarkers both in patient care as well as in drug development to improve efficiency and improve care. So why are biomarkers important? In the clinical setting, biomarkers allow us to diagnose disease, which is the foundation of therapy. They allow us to monitor the disease process. So you can determine if a patient is getting better or getting worse and adjust their therapy accordingly. They're used to monitor response and toxicity to therapies. So you can determine if a patient has too high or too low of a dose potentially, and then go on to adjust that dose to have an appropriate therapeutic window for that patient. Now, in the therapeutic product development setting, biomarkers can inform the safety and pharmacologic activity in early non-clinical studies of a drug or of a candidate drug. They can be used for proof of concept in early clinical studies to demonstrate that your drug is getting to the target site and having some sort of action. They can be used to determine doses for later confirmatory efficacy trials. They can be used to actually demonstrate efficacy in later phase studies if it's a surrogate biomarker, which I'll discuss in more detail in the next section. And also they can be used to demonstrate toxicity in all stages of drug development. So despite the widespread use of biomarkers, until recently there was not a well-accepted standardized definition for biomarkers or categories of biomarkers for their use. So a couple of years ago, the NIH and FDA collaborated to create a biomarker working group, and they created what they call the BEST resource. And this stands for Biomarkers, Endpoints, and Other Tools resource. And what this is a glossary of terminology and uses of biomarkers and endpoints in basic biomedical research, medical product development, and clinical care. And this is a freely, publicly available resource. It's available at the link that's on the slide that's currently being shown. So you can go and access that, and it's actually meant to be a living document. So it's going to be updated from time to time as the field evolves. So BEST defines biomarker as a defined characteristic that is measured as an indicator of normal biological processes, pathogenic processes, or response to an exposure or intervention, including therapeutic interventions. And they recognize 5 different types of biomarkers, including molecular, so this could be a genotype of a patient, histologic, so this could be information from a slide from a tumor biopsy, radiographic biomarkers, such as imaging biomarkers, again, very useful in oncology as well as other settings, and physiologic characteristics. So this could be something such as blood pressure. So this slide has several examples of biomarkers. These are what are considered traditional or standard biomarkers. You've probably all heard of all of these. Things such as blood glucose and cholesterol are measured in medical practice as well as used in the drug development setting. Things such as transaminases, bilirubin, and ALK-Phos are used to assess liver function. Serum creatinine clearance, and cystatin C can all be used to assess kidney function. And again, these are traditional biomarkers that have been found acceptable over time through clinical experience and weight of evidence. So they're well accepted in many different settings. People aren't really going to question their use. They've got many different uses. However, not all biomarkers are going to have this level of data and this history of use in the medical setting. So there's a need to develop standards to standardize these biomarkers so that they can be used within certain contexts in drug development. And I'll discuss that in the third section today. So the last slide showed many different types of standard biomarkers, but I want to point out that a biomarker can take on many different forms. So this is a slide with different biomarkers for cardiovascular disease, and organizes them into several different categories based on what they're used for. So they recognize functional biomarkers, and these can be things such as exercise testing or assessments of endothelial function or vascular function. They recognize quality of life biomarkers. So this could be the results of a patient-reported outcome questionnaire, could be considered a biomarker. A medical device-derived biomarker, so information that's gained from things such as implanted ICDs or sensor patches can be considered a biomarker. Imaging biomarkers, again, echocardiography or MRI can be biomarkers. And lastly, molecular biomarkers. And this, again, is the category that's probably gotten the most attention recently due to advances in technologies. These are things such as proteins, peptides, mRNAs, or genetics. They can be measured in blood or urine samples, and these can also be very useful biomarkers. Similarly, this slide shows different potential biomarkers for prostate cancer, and this is organized by where the biomarkers originate. So the imaging biomarkers such as transrectal ultrasound or CT or MRI or PET imaging. Serum biomarkers, so you can look at circulating tumor cells in the serum, as well as molecular analyses in DNA and RNA. The tissue biopsy biomarkers, so histopathology information, Gleason score, immunohistochemistry, or you can do molecular analyses actually on the tumor tissue to look for somatic mutations and changes in gene expression. And lastly, urine biomarkers. So there's many different metabolites that are useful to measure in urine for prostate cancer patients. So biomarkers can take on a very broad array of things, not just the traditional biomarkers that you've likely heard of, such as blood pressure and glucose. So BEST recognizes 8 different categories or categories of biomarkers, and they can be used for many different things. So susceptibility biomarkers can inform whether or not you'll develop a disease. An example of this would be BRCA mutations for breast cancer. They can inform the lifetime risk of a patient for developing breast cancer. Diagnosis of disease, so do I have an actual disease? So glomerular filtration rate in chronic kidney is diagnostic of chronic kidney disease. Prognosis, so how will I live longer? PSA changes can be informative of prognosis for prostate cancer. Prediction, will I respond to treatment? So certain BRAF mutations for some melanoma treatments can inform your likelihood of response to that drug. So if you have that predictive mutation, that might be a good drug for you, whereas if you don't have that predictive mutation, then the drug, you're unlikely to respond to that drug, and therefore a different line of therapy would be more appropriate. So that's a predictive biomarker. Response, so did the treatment work? So an example of this would be INR for response of warfarin in the treatment of stroke or in the reduction of risk of stroke. So INR within a certain window is informative of a likelihood and a decrease of your risk for stroke. So this can inform whether or not the treatment is working appropriately. Monitoring, has the condition changed? HIV RNA levels are commonly measured in HIV and AIDS patients to monitor disease process. And safety biomarkers, so am I having an adverse event? So ALT levels can be informative of whether hepatotoxicity is occurring at a very early stage, and therefore you can monitor this as a safety biomarker and potentially stop the drug or alter the dose to prevent worse hepatotoxicity from occurring. And lastly, I'd like to just note that these functions are not mutually exclusive, so there's much overlap in these. So for example, I used INR as a response biomarker for warfarin and the reduction of the risk of stroke. However, this is also considered a monitoring biomarker because patients that are taking warfarin, they monitor their INR regularly to make sure that they're in the appropriate therapeutic window. And it could also even be considered a safety biomarker because if INR goes above a certain level, it increases your risk for bleeds, and therefore it could be considered a safety biomarker for that drug as well. So these are not mutually exclusive. Now that I've discussed the many different categories of biomarkers, I want to focus in a little bit more narrowly on the ones that could be considered efficacy biomarkers or biomarkers of drug effect. So the first I want to talk about is a PD, or response biomarker. So this is a biomarker used to show that a biological response has occurred in an individual who has been exposed to a medical product or an environmental agent. An example of this would be sweat chloride, can be used as a pharmacodynamic or response biomarker when evaluating patients with cystic fibrosis. And it's used to assess the response to cystic fibrosis transmembrane regulator potentiating agents. Also, hemoglobin A1C may be used as a pharmacodynamic or response biomarker when evaluating patients with diabetes to assess response to antihyperglycemic agents or lifestyle changes. And I chose these two examples specifically because the sweat chloride could be considered more of a true PD biomarker. We haven't actually approved any drugs based on this endpoint in clinical trials yet, but it is very informative of whether a CFTR potentiating agent is having some activity in a patient. On the other hand, hemoglobin A1C is a little bit more established. This biomarker has been actually used to approve some drugs, some antihyperglycemic agents, so it could actually be considered a surrogate biomarker. Safety biomarkers, so just to point out, safety biomarkers can also be a biomarker of an effect. It's just a biomarker of a negative effect. So BEST defines a safety biomarker as a biomarker measured before or after an exposure to a medical product or an environmental agent to indicate the likelihood, presence, or extent of toxicity as an adverse effect. A couple of examples of these would be hepatic aminotransferases and bilirubin may be used as safety biomarkers when evaluating potential hepatotoxicity. And neutrophil count may be used as a safety biomarker when evaluating patients on cytotoxic chemotherapy. And they can be used to adjust dose, determine the need to interrupt therapy, or consider the use of growth factors. So they can be an early safety signal that you can use to monitor a patient and adjust the medication accordingly to prevent a worse toxicity from occurring. Surrogate endpoints. So BEST defines surrogate endpoint as an endpoint that is used in clinical trials as a substitute for a measure of how a patient feels, functions, or survives. And it actually recognizes 3 different categories of surrogates based on the quantity and quality of evidence that supports their use as a surrogate. So the first is a validated surrogate endpoint. This is an endpoint that's supported by a clear mechanistic rationale and clinical data providing strong evidence that an effect on the surrogate endpoint predicts a clinical benefit. Therefore, this can be used to support traditional drug approval without the need for additional efficacy information. So this can be a standalone endpoint in an efficacy trial for a drug. The next category is reasonably likely surrogate endpoint. This is an endpoint supported by clear mechanistic and/or epidemiologic rationale, but insufficient clinical data to show that it is a validated surrogate endpoint. So in this case, we have a little bit less data indicating that it's a valid surrogate, and therefore these endpoints can be used for accelerated approval of drugs, for expedited access for medical devices. And I'll cover the difference between traditional and accelerated approval a little bit more in depth in the next section. But a reasonably likely surrogate can be used for accelerated approval purposes. The last category is a candidate surrogate endpoint, and this is an endpoint that's still under evaluation for its ability to predict a clinical benefit. So this could be anything that has some data showing that it's a potential surrogate, but not enough data to truly be able to know how it can effectively be used or appropriately be used. Next, I'd like to cover some examples. So an example of a validated surrogate endpoint would be blood pressure reduction is a validated surrogate endpoint for reduction in rates of stroke, myocardial infarction, and mortality, and has been used for the basis of approval for drugs intended to treat hypertension. So this slide shows the example of some data that can be used to demonstrate that a surrogate is a validated surrogate. And it has each of these dots on the slide actually is a clinical trial endpoint, not an individual patient. And they show that across all these different clinical trials, which many of these are many tens of thousands of patients, overall I believe the representation is over 500,000 patients included in these clinical trials. And they show that there's a relationship between, on the x-axis, the blood pressure reduction that was observed in these trials, and on the y-axis, the odds ratio of a cardiovascular event. So you can see across multiple different trials that had multiple different drugs from multiple different drug classes, we see that this blood pressure lowering effect reduces our risk of cardiovascular events. And this is great data to show that this is a validated surrogate endpoint for antihypertensive agents. Now, an example of a reasonably likely surrogate endpoint, there's a couple of these on this slide, including radiographic evidence of tumor shrinkage, so this would be response rate, as well as progression-free survival. In certain cancer types have been considered reasonably likely to predict an improvement in overall survival with certain therapies, and therefore they've been used to support accelerated approval of drugs to treat these cancer types. So in some cancers, but not all cancers, we consider radiographic evidence of tumor shrinkage to be a reasonably likely surrogate. Second example is outcomes of 6-month follow-up treatment, so sputum cultures and infection relapse rate have been considered reasonably likely to predict the resolution of pulmonary tuberculosis and have been, again, used to support accelerated approval of drugs to treat tuberculosis. And there's no graph showing all the different data on these ones because, again, if there was that sort of data supporting it, it would be considered a validated surrogate. There's not quite as much data, so it's considered a reasonably likely surrogate for these two examples. And I won't cover candidate surrogate biomarkers because, again, these could be anything with some data indicating that it's a potential surrogate, but not enough that we know how we can effectively utilize them. So now that I've covered the definitions and utility of biomarkers, I'd like to go over using biomarkers in drug development and how they can be effectively utilized in clinical trials. This slide shows the outcomes of a study that was conducted a few years ago at AstraZeneca, and what they did was they looked over their product portfolio for the previous few years, and they tried to determine what the key contributors to drug development success were. And the things that they identified as identifying successful projects as opposed to projects that did not make it to market or projects that were considered failures were having the right target, the right tissue, the right safety profile, the right patients identified for the drug, and the right commercial potential. And what I want to point out here is that in order to determine these things, you've got to be able to have some kind of data to support whether or not you have the right target, the right tissue, the right safety profile, et cetera. And all biomarkers can be utilized to inform virtually all of these things. And I've underlined on the slide the examples where they specifically mention having biomarkers to determine these things, but it's not really limited to those. There's many of the other things that are listed on this slide can be informed by appropriate use of biomarkers. So this is why biomarkers are considered so important in drug development setting. I'm not sure what experience everybody has with clinical studies, but to discuss the use of biomarkers throughout drug development, we need to kind of make sure that we're on the same page as far as the different phases of clinical studies. So I briefly want to cover that on this slide. So phase 1 clinical studies are the initial introduction of an investigational new drug into humans. These are frequently in healthy subjects, and the goal of these is to obtain some pharmacokinetic, pharmacodynamic, and safety information to design scientifically valid phase 2 studies. Now, your phase 2 studies are your early controlled studies to get information on the drug's effectiveness in the target patient population. So here we're moving into patients. Studies are slightly larger usually, and they're in the actual target patient population to determine if the drug is having the appropriate effect. And lastly, phase 3 studies are your adequate and well-controlled studies to gather information about effectiveness and safety that is needed to evaluate the overall benefit-risk relationship for the drug for marketing. So overall, your goal here is to demonstrate substantial evidence of effectiveness and a positive benefit-risk profile in order to get the drug on the market. So how are drugs approved? Under the Food, Drug, and Cosmetic Act, it states that the FDA cannot approve an application to market a drug unless there is substantial evidence that the drug will have the effect it purports to have. And this evidence of efficacy is primarily derived from adequate and well-controlled clinical trials that demonstrate improvement based on a clinical endpoint that directly measures how a person feels, functions, or survives. So that's a very important concept. These feels, functions, or survived endpoints are considered clinical endpoints that are often referred to as hard outcomes in clinical studies. So if you improve how a patient feels, such as reduction in pain, how they function, such as improvement in activities of daily living or 6-minute walk test or something of that sort that's a measure of function, or how long they survive, those are considered clinical efficacy endpoints and are generally the basis for drug approval. However, there are exceptions built into this where well-established surrogate endpoints can be used to approve a drug. However, this term is not defined, but this would kind of be the category of the validated surrogate endpoints under BEST, and reasonably likely surrogate endpoints under the accelerated approval program. So the accelerated approval program is meant to address diseases that are serious medical conditions where there's an unmet medical need. So a patient population that doesn't have any drugs available with a serious disease, they allow reasonably likely surrogates to be the basis of approval under the accelerated approval program to get those patients the drug. However, this does require a confirmation study. So in the post-market setting, they have to do a confirmation study on a feels, functions, or survives clinical endpoint in order to maintain marketing for that drug. So how can biomarkers be used to inform drug development? They can be used in virtually every different stage. So even back in the preclinical studies and your basic clinical or your basic research when you're trying to identify pathways and identify potential drugs, they can inform drug target selection. So what pathway you want to target for a certain disease. They can inform the drug's mechanism of action. Once you get into clinical studies, they can be used in phase 1 through 3, as well as even in post-approval studies to stratify patients. So if there's a predictive or prognostic biomarker that you think might affect your outcomes and you want to make sure that you have equal numbers of patients in each group, in your drug group and in the placebo or in the comparator group, you can stratify patients, and this can eliminate some noise from your clinical studies. They can be used for safety assessments, for patient selection and enrichment, to help determine the appropriate dose selection, as well as, again, in efficacy assessments. So how are efficacy biomarkers used in drug development? Basically, they're used to measure response to a treatment, and this can take on several different forms. In your earlier phase studies, they can be used to establish a biological effect. This would be a use of a pharmacodynamic biomarker in early trials, and this basically shows target engagement. Your drug is getting to the target and it's having some sort of effect that you consider desirable. They also can be used to determine suitable doses and regimens to carry forward into later phase studies. And lastly, they can be used to replace health outcome endpoints. And this again would be the surrogate endpoints in later phase trials. The idea here, the promise of using surrogate biomarkers is that they promise to shorten clinical trials and reduce trial size. And the way that they do this is that generally your surrogate biomarker, the change in that is going to occur at a time point before the actual clinical endpoint is going to occur. So you can have a shorter trial. They can also be used to reduce trial size, because generally a surrogate biomarker is going to have less noise associated with it than a true clinical endpoint. So as a survival endpoint is very hard to demonstrate, because not only is your drug having an effect on that, but the patient's lifestyle and things of that sort are also impacting the clinical efficacy endpoint. So therefore a surrogate biomarker can be used to reduce the trial size, because there's going to be less noise surrounding that endpoint. So shorter clinical trials and smaller clinical trials can be much more efficient, because the larger and the longer clinical trials get, the more expensive they are and the harder they are to conduct. And also, again, the basis for accelerated approval under the accelerated approval program, and also can often support full approval in the case of validated surrogate endpoints. Here's an example of the use of biomarkers to determine dose. So this is the drug belimumab, which is a B lymphocyte stimulator inhibitor. So it exerts its effect through lowering B cell counts. And in this case, they looked over multiple different B cell subsets, and they looked over a couple of different doses, and they used this data to determine that the higher dose was having a greater effect on the B cell subsets, and they used this information to help inform their dose and ultimately the approval of the drug. So how are safety biomarkers used in drug development? Safety biomarkers are used to identify and monitor potential drug-related toxicities. So in early phase animal studies, you can conduct toxicogenomic studies to evaluate safety. They can also be used to help determine the starting dose for first-in-human trials. So this is calculated from the No Observed Adverse Effect Level, or NOEL, in toxicology studies, and biomarkers can be used to inform this. And then you base your first-in-human starting dose on that. They can be used to identify, monitor, and avoid toxicities in clinical trials. And they can, again, also be used to determine the appropriate dose. So you want to have a dose below when you start seeing toxicity. And in some cases, they can actually be used to inform dose reduction schemes. So this is very common, particularly in oncology and in diseases where we dose at the very high end of the dose response curve, where because the disease is very severe, we're trying to squeeze every bit of efficacy out of that drug, and we're willing to tolerate some safety or adverse events. And therefore, you can use biomarkers to monitor, or safety biomarkers to monitor potential toxicities. And before you have a clinical issue occur, you can adjust the dose or have a dose interruption for that drug. So here we have a very simplistic view of drug effects. And when we're trying to determine which biomarker we want to look at in drug development, the simplistic view would have the drug interacting with the target. This then leads to some change in a biomarker, which then is predictive of your clinical outcome, so how the patient feels, functions, or survives. However, biology is rarely that simple. So this is a more realistic view of how biomarkers change in response to a drug. So here you have the drug interacting with a target. This myriad of changes occur in different biological pathways. Some are in the causative pathway, some might be parallel, some are in sequence with each other. And then the changes in these biological pathways ultimately produce some change in clinical outcome, and again, how a patient feels, functions, or survives. So any of these green or red dots here could be a potential biomarker that we would measure in drug development to determine if our drug is having an effect. And therefore, we need to decide which one or ones we want to look at. And the basis of this can be based on what we actually want to show depending on where we're at in drug development. And I'll cover that over the next couple of slides. So here's an example of efficacy biomarker for an ACE inhibitor. So here I have the renin-angiotensin-aldosterone pathway, and this is also, again, a simplified look at that, but most of the important components are here. So in the renin-angiotensin-aldosterone pathway, we have angiotensinogen is converted by renin into angiotensin 1. The angiotensin converting enzyme then converts angiotensin 1 into angiotensin 2, which acts via the angiotensin 2 receptor to cause aldosterone secretion, sodium and fluid retention, sympathetic nervous system activation, and vasoconstriction. This leads to an increase in blood pressure, and this causes hypertension. So hypertension, although recognized as a disease, is also in itself kind of a biomarker or a surrogate of increased risk for stroke and cardiovascular events. So ACE inhibitors are drugs that inhibit this angiotensin converting enzyme from converting angiotensin 1 into angiotensin 2. So anything downstream of here could be a potential biomarker to show efficacy or drug effect with an ACE inhibitor. So one potential biomarker, assuming that there was an analytically validated way to measure it, would be angiotensin 2. So the advantage is that angiotensin 2 is very proximal to the drug target. However, it's very distal from the clinical outcome. So this would make it a candidate biomarker to demonstrate drug activity. However, it makes it a poor candidate as a surrogate biomarker. And the reason for this is because it's very proximal to where our drug is having its action. It's actually the product of the enzyme that we're inhibiting. It can be very sensitive to inhibition of that enzyme. However, because it's so far from our outcome of hypertension, a lot of different things have to happen before the drug actually exerts that clinical effect. And a lot of compensatory things can kick in and other things can happen that actually prevent the drug from ultimately having the effect on hypertension. So it would be a poor surrogate outcome because so much has to happen before that clinical efficacy actually occurs, but it's a very sensitive biomarker to demonstrate drug activity. So we could use this in our early studies as a pharmacodynamic biomarker to show that our drug is getting to the target site of action, potentially inform doses for our later phase studies. Now, alternatively, we could look at blood pressure as a biomarker of effect for an ACE inhibitor. Now, blood pressure, in contrast to angiotensin 2, is very distal from the drug target, but it's very proximal to the clinical outcome. This makes it a good candidate surrogate biomarker. However, it's a problematic biomarker for demonstrating drug activity. And the reason for this is, again, because so much has to happen between the inhibition of the enzyme and the change in blood pressure that a lot of compensatory things can kick in, and it's going to take a larger study with more patients to show an effect on this biomarker. So when you're in an early phase study, you might not want to invest that much into a very large study with a lot of patients to demonstrate that it has a change in blood pressure. So you might instead monitor angiotensin 2 again as a pharmacodynamic biomarker. Whereas when you get to your later phase studies and you're trying to truly show an effect, this would be a candidate surrogate biomarker to look at in your later phase efficacy studies. So returning to our simplistic view of drug effects, after the drug interacts with its target, we have a choice of many different biomarkers that might change in response to that are ultimately, we hope, predict clinical outcome. But where that biomarker is and what properties it has can affect how we want to utilize it for drug development. Now that I've covered the different types of biomarkers and pharmacodynamic biomarkers versus candidate surrogate biomarkers, I want to discuss the issues associated with using surrogate biomarkers in drug development programs. So again, surrogate endpoints are defined as an endpoint that is used in clinical trials as a substitute for a direct measure of how a patient feels, functions, or survives. Different factors that should be considered when you're deciding whether or not to use a surrogate endpoint would be biological plausibility, its success in previous clinical trials in predicting outcomes, and the risk-benefit and public health considerations of the drug. So this isn't actually related to the biomarker, but it's more of a public health consideration and a standard of care, how the medical practice is currently occurring in the patient population that you're interested in. So for biological plausibility, things that favor the use of surrogate include epidemiological evidence that's extensive and consistent across studies, a quantitative relationship, a credible animal model to show drug response, and a well-understood disease pathogenesis and drug mechanism of action, and also, again, the surrogate being relatively late on the biological path. Things that do not favor surrogates in terms of biological plausibility are basically all of the opposites of that. So inconsistent epidemiology, no quantitative epidemiological relationship, no animal model, unclear pathogenesis and unclear drug mechanism, and the surrogate being remote from the clinical outcome. As far as clinical trial success, so effect on a surrogate has predicted outcome with other drugs of the same pharmacological class. This can be supportive of it being a surrogate for another drug in that same class. Whereas if there's an effect on a surrogate has predicted outcome in several different classes, this supports more general use. So this would be the difference between, say, for an antihypertensive drug, if you had a diuretic and it had only been shown to predict outcomes with other diuretics versus if it had been shown to predict outcomes with other classes such as ACE inhibitors and calcium channel blockers and other antihypertensive agents, that would support more general use for another antihypertensive agent from another class. Things that don't favor the use of surrogates in clinical trials would again be the opposite, so negative outcomes in clinical trials without a clear explanation for that, as well as inconsistent results across drug classes. So the last factor here that is discussed is the risk-benefit and public health considerations. So in cases where you have a disease that's serious or life-threatening with no alternative therapies, you're going to be more likely to accept a surrogate for that type of a disease because the stakes are, we really want to get patients those drugs, and therefore we're willing to accept a little bit of uncertainty for the efficacy because those patients have no other alternatives available to them. A large safety database, again, if the drug has been shown to be very safe and we're confident in that, it's more likely to use a surrogate. Short-term use of the drug, as well as if there's difficulty in studying the clinical endpoint. Things that do not favor the use of a surrogate in terms of public health considerations are a non-serious disease, and if there's alternatives available for patients with that disease. Little safety data, so if we're not confident in the safety profile, we want to see improvements in those hard outcomes or clinical efficacy endpoints. Long-term use of the drug, and if the clinical endpoint is easy to study, there's no reason to use a surrogate instead. You can just study the clinical outcome. And lastly, if there's a long delayed small effect in relatively healthy patients, this would not support the use of a surrogate. So the next few slides here are adapted from a paper by Dr. Thomas Fleming, who has spoken extensively on the subject of surrogates. If you're interested in surrogates at all, I highly recommend that you read the reference there, and he has a couple of other papers which are fairly easy to identify or to find as well. He has many different informative papers on this subject, but I'll attempt to go through some of the main considerations that he lists and the cautions for use of surrogates. And the reason that we often have to be cautious is because there's a lot of candidate surrogate biomarkers that are biologically plausible and they seem like they're sure to predict the clinical outcome. However, there's reasons that they often fail to end up being validated surrogates, and several of these are discussed over the next couple of slides. The first one, so in the first case here at the top of the slide, we're discussing the transmission of HIV from mother to child. So in this case, we have HIV viral load is causative of that transmission of HIV from mother to child. However, many things are going to also be correlated with HIV viral load, such as CD4. So if you have a biomarker such as CD4, it's also going to look like it's associated with the transmission. However, if we have a drug that affects our CD4 levels without impacting HIV viral load, it's not going to affect that transmission. The example at the bottom of the slide is fairly similar. So tumor burden is causative of cancer symptoms and death. But there are many biomarkers that are going to be associated with tumor burden, such as CEA or PSA. So if we were to have a drug that were to affect those biomarkers, CEA or PSA, without actually impacting the tumor burden of the patient, it's not going to go on to change the cancer symptoms or likelihood of death in those patients. So if it's correlated with a true biomarker, or if it's correlated with the clinical outcome, but it's not in the causative pathway, it could lead to it not being a good surrogate. The second slide for caution of surrogates here discusses a couple of different issues. So the example on top, in this example is discussed in detail in the paper. However, it discusses the patient population that's post-myocardial infarction and the use of thrombolytic agents. So in this example, they discuss, they have a novel thrombolytic agent that in a smaller phase 2 study, it had a better impact on what they were considering as a surrogate outcome of TIMI-3 blood flow, which is a measure of blood flow to the myocardium, which is considered a surrogate for 30-day mortality. So their novel drug had a better effect at, I believe it was the 60 and 90-minute endpoint or time points on TIMI-3 blood flow. So their hypothesis based on that was that this would lead to a better 30-day mortality outcome with their novel drug. However, when they ran their larger phase 3 study, they showed that it was actually numerically worse than the older agent, and that it did not, in fact, go on to improve 30-day mortality. So they investigated the reason for this and possible causes, and one thing that they discovered was that in that initial phase 2 study, The novel agent actually had inferior improvement in TIMI3 blood flow at the 30-minute time point. So perhaps having that earlier restoration of blood flow was actually more important than the greater magnitude at the later time points. And therefore, this was unable to predict 30-day mortality. So even though it seemed like a great potential surrogate outcome, not measuring it at potentially the correct time point led to this not being a validated surrogate for this disease. And because they measured the clinical outcome, they were able to determine this. The example on the bottom kind of gets at the opposite situation, and that is by using a surrogate, you can sometimes potentially lead to the conclusion that your drug does not have an effect, when in fact it does. So they discuss a study in chronic granulomatous disease, and they were using an interferon gamma as an agent, and they considered using bacterial killing to prevent as a surrogate for recurrent serious infections. They ended up actually looking at the clinical outcome instead, and that was a good thing because interferon gamma did not have an effect on bacterial killing, but actually did have an important effect on recurrent serious infections. So the case here is that you have actually multiple causal pathways that are leading to the clinical outcome of recurrent serious infections, and interferon gamma affected one of these that was not captured by the surrogate, or the potential surrogate of bacterial killing. So again, it can also lead to a false conclusion that your drug does not have a positive effect, when in fact it does. And the last slide, the last figure in this paper that I want to cover is, this discusses the situation where you have an intervention that has an effect on a biomarker which you believe predicts a true clinical endpoint. However, the drug might also have off-target effects that could then cancel out the benefit that you're observing based on the biomarker. So an example of this would be, to stick with hypertension, since I've discussed that in a few different ways, a diuretic might have a positive impact on the biomarker of blood pressure, which predicts the true clinical endpoint. However, if it caused a lot of hypokalemia, which is a known side effect of many diuretics, this could potentially cause negative effects, which would then cancel out the true clinical endpoint benefit. So I've discussed in detail a lot of aspects of surrogate endpoints as well as pharmacodynamic endpoints. So to quickly summarize these and summarize the take-home message, surrogate endpoints are important in drug development because they can increase clinical trial efficiency. It does this via shorter duration of trials because the change in the surrogate will generally occur at an earlier time point than the change in the clinical efficacy outcome. And it can do this using fewer patients, so you can power your trial with less patients to show the benefit that you want to show. This really leads to much more efficient drug development. Factors that favor surrogate use include biological plausibility, success in predicting outcomes for multiple drugs and drug classes, and unmet medical needs for the patient population. However, caution is generally advised when using surrogates because a lot of candidate surrogate endpoints do not end up being great predictors of the clinical efficacy outcome. And again, many surrogate biomarkers are ultimately shown to be poor surrogates. So before I move on to the next section, I want to briefly discuss the considerations for measuring biomarkers. So measuring biomarkers is often overlooked, but this is extremely important. So whether we're in the clinical care setting where we're making judgments on how we're going to treat a patient, or whether we're in the drug development setting where we're making decisions as to whether or not to continue investigating a drug for a certain patient population, measuring the biomarker accurately is extremely important because these are important decisions and we want to make sure that we're accurately measuring and that we're doing what we think we're doing before we use those data to make those decisions. So appropriate sample collection, processing, and storage are all critical to ensure validity of results. And appropriate can mean different things in different situations. So whether you're measuring RNA versus DNA, you might have to have different procedures in place for your sample collection and processing and storage. Whether you're measuring something in blood versus urine, for instance, can have an effect on what appropriate sample collection and processing and storage is. Analytically validated assays should be used for quantification of biomarkers that are used for drug development and/or regulatory decisions. And again, so going back to the BEST definition, BEST defines analytical validation as establishing that the performance characteristic of a test, tool, or instrument are acceptable in terms of sensitivity, specificity, accuracy, precision, and other relevant performance characteristics using a specified technical protocol, which may include the specimen collection, handling, and storage procedures. So just to point out, this is validation that the test, tool, or instrument's technical performance. It is not validation of its usefulness. So again, this is simply you're validating the measurement of the biomarker, that you're measuring what you think you're measuring. It doesn't necessarily mean that just because you're measuring a biomarker that it's the appropriate biomarker to use in a certain situation. It just means that the analytical data are going to be reliable. So the last thing that I would like to cover briefly is the Biomarker Qualification Program, which is currently in the Center for Drug Evaluation and Research. And the website link is on this slide that's being shown currently. If you are interested in the biomarker qualification process, there's much more information on the website there. I'll briefly discuss the highlights. So in many cases, standard biomarkers are not optimal, and other biomarkers may offer better attributes. However, every year there are hundreds of new biomarkers that are reported in the literature that are potentially have new useful, new uses in drug development or in clinical practice, or might simply be a better indicator of some process than the current gold standard. But how do we know which ones are actually valuable for use? And how do these novel biomarkers become the gold standard instead of the ones that we're currently using? So historically there have been many different biomarker acceptance pathways. Probably the most common one throughout history is simply scientific community consensus. So if there's a biomarker that's a candidate and there's many published articles commenting on that establish its utility, ultimately this will sometimes lead to guidances by the FDA and regulatory acceptance in the drug development process or even in the patient care process. But this can often take a very long time to establish, and many different studies need to be conducted. The drug approval process can use to acceptance of a biomarker, but this is generally more narrow. So within a certain drug development program, they might use a certain biomarker, and that's considered acceptable in that context. And this could make it into drug labels and in the reviews that are conducted by the review staff. And sometimes this will ultimately make it into guidance. However, there's often uncertainty over how exactly this can be appropriately used. And more recently, there's been the Biomarker Qualification Program. So this is a formal process for biomarker qualification. There's several different guidances that are produced on this. There's actual formal FDA review of the biomarker for its utility. And the program often conducts workshops to discuss the need for the standardization of data generation and the data that's needed to qualify a biomarker within a certain context of use. So what exactly is qualification? Qualification is a conclusion that within the stated context of use, the biomarker can be relied upon to have a specific interpretation and application in drug development and regulatory review. So what the context of use means is it's a comprehensive statement that fully and clearly describes the manner and purpose of use for the biomarker in drug development. Qualification is a formal regulatory review and acceptance process of the biomarker that reaches across all FDA and CDER review divisions. So once a biomarker has been qualified, the different review divisions won't treat it differently. It's qualified for that specific context of use and can be used across all review divisions. Qualification results in the scientific acceptance and regulatory certainty, and this is based on a weight of evidence argument that's presented in the qualification process. Now, what exactly is that process? Here we have the biomarker qualification roadmap. The process starts with a letter of intent. This initiates the qualification process of a biomarker for a proposed context of use in drug development. Next comes a qualification plan. This defines the development of to generate the necessary supportive data to qualify the biomarker for the proposed context of use. After the data are generated, the full qualification package is submitted. This contains all accumulated data to support the qualification of the biomarker for the proposed context of use. And lastly, there's a qualification recommendation. So this contains the FDA's determination on whether the biomarker is qualified for the proposed COU based on a comprehensive review of the full qualification package. So this is somewhat analogous to drug review, except you're reviewing the utility of the biomarker within its specific context of use, and it comes to a determination of whether the data are appropriate to support the qualification of the biomarker for whatever the context of use is. And the context of use can be very broad. It can be everything from a toxicity marker for an animal study to an enrichment biomarker for clinical studies, or it could potentially be a surrogate endpoint, which we've discussed. It could be qualified for use. So this slide I don't want to discuss in a ton of detail. However, it's a list of the biomarkers that have currently been qualified. It's fairly short currently, but there are many programs in development as of right now, and the rate at which we're qualifying them is actually increasing. So I just want to point out that, again, the diversity here, so there's non-clinical biomarkers as well as clinical biomarkers, and I believe the first one that was qualified is the first one on the page here, which is drug-induced nephrotoxicity biomarkers. This is in preclinical studies. More recently, things that have been qualified are prognostic biomarkers for enrichment of clinical trials within dominant polycystic kidney disease. So there's a broad range of things that can be qualified for use, and this is really driven by what the bottlenecks in drug development can be. So if there's something where it currently takes a whole lot of data to demonstrate that something is safe or something is effective in a certain patient population, then different groups can focus in and try to develop biomarkers that can solve those problems more efficiently. And this is often done, this can be done by anybody, I would also like to point out. So it can be an individual company could do this if they wanted to. It's often done by consortia, so groups of either academics or industry groups can get together and pool data to try to make an effort to qualify a biomarker to help make the drug development process more efficient for all of them. So to summarize and provide some conclusions for the entire lecture, biomarkers are essential to the practice of medicine as well as the development of medical products, including drugs. In drug development, biomarkers of effect and safety are used to demonstrate proof of concept, select appropriate doses, and demonstrate efficacy. Caution is warranted when using surrogate endpoints, and the biomarker qualification program can improve acceptance of biomarkers within a specific context of use. So with that, I would like to say thank you very much for listening. I hope this has been beneficial to everyone.